Imagine this. A patient with a failing liver walks into a hospital. Instead of getting on a year-long transplant list, the doctors print a new liver, layer by layer, from the patient's own cells. Sounds like an episode of Black Mirror. Well, it's not. Thanks to groundbreaking advances in 3D printed tissue engineering, scientists are now creating collagen scaffolds that grow real, functional human tissues. This could revolutionize everything from organ transplants to wound healing, and even how we test new drugs. The problem with organ transplants. You may know someone who's needed an organ transplant, maybe a kidney, maybe a heart, and quite possibly, maybe it was you. Here's the hard truth. There are over 100,000 people in the US alone waiting for transplants. Not to mention every day, 17 people die because the organ they need just isn't available. These are more than just numbers. They're fathers, daughters, neighbors, and coworkers. Real, everyday people with lives on hold, families on edge, and hope thinning. Unfortunately, even when a donor organ does come through, miraculously, and sometimes tragically, it's not a guaranteed fix. There's the risk of the recipient's immune system launching an all-out attack on the new organ, treating it like an invader. Now the person is saddled with a lifelong dependence on immunosuppressants, which themselves can bring a multitude of side effects, infections, increased cancer risk, and not to mention an overall weakened immune response. It's obviously far from ideal. And there's the darker side that people prefer not to talk about. The global black market for organs is thriving, preying on desperation and the high cost of transplants. Organ trafficking rings exploit vulnerable people, creating a gruesome economy around something that should be rooted in compassion and care. But what if we could change the equation entirely? What if the organ didn't come from someone else? What if it came from you? Imagine printing the scaffolding of a heart or liver tailored precisely to your body, then growing your own cells on it. No immune rejection, no sketchy underground markets, just science, doing what it does best, solving problems that once seemed unsolvable. 3D printed tissue engineering, a game changer in regenerative medicine, potentially redefining the future of transplants. Let's break this down. At its core, 3D printed tissue engineering is about merging biology, engineering, and cutting-edge printing technology to recreate living functional tissues outside the human body. It's part science fiction, part science fact, and entirely groundbreaking. Think of it like baking, but with cells instead of flour. You need three main ingredients. 1. A structural framework called a scaffold, which gives the tissue its shape and mechanical strength. 2. Living cells referred to as bioinks, which are the actual living components that do all the growing, healing, and functioning. And 3. Nutrients, the biological fuel to keep those cells alive and thriving as they grow into the structure. Using ultra-precise 3D bioprinters, scientists build this scaffold layer by layer, carefully mimicking the incredibly intricate structure of human tissues. We're talking about the microscopic geometry of things like blood vessels, cartilage, skin, even heart muscle. The scaffold isn't just a mold, it's a blueprint, a lattice that guides the cells to grow in the right places and in the right ways. Once the scaffold is printed, scientists seed it with human cells. These cells attach, multiply, and if the environment is just right, start acting like they do inside your body. They connect, communicate, and organize themselves into functional tissue. It's not just about making tissue that looks like the real thing. It has to act like it too. That's where collagen, your body's natural building block, steps into the spotlight. Strong, flexible, and biocompatible, it's the perfect foundation for growing new life. If our bodies were buildings, collagen would be the rebar. It's the most abundant protein in the human body, woven throughout our skin, bones, tendons, cartilage, and connective tissues. It holds us together, quite literally. Think of it as the scaffolding nature uses to build and maintain the human form. It's strong yet flexible, tough yet adaptable. And here's the best part. It's naturally biocompatible. That means the body recognizes it, accepts it, and integrates it. No alarms, no rejection. Because of all this, collagen has huge potential in medical applications, especially when it comes to regenerating or replacing damaged tissue. But there's a catch. It's notoriously difficult to 3D print. Why? Well, collagen is delicate. It's a soft, fibrous material that doesn't hold the shape the way hard plastics or metals do. It tends to collapse, warp, or degrade during the printing process. It's like trying to build a skyscraper out of cooked spaghetti, technically possible, but incredibly frustrating. And for years, that was the bottleneck, a major hurdle that kept scientists from using collagen in 3D printing. Until recently, that is. The breakthrough. In a stunning leap forward, researchers at Harvard's West Institute cracked the code. For years, scientists struggled with how to 3D print collagen, without it turning into a saggy mess. But then came a game-changing technique known as fresh, short for freeform, reversible embedding of suspended hydrogels. It might sound like a mouthful, but the concept is brilliant and wildly effective. Let's translate that into layman terms. Instead of printing collagen into open air where it collapses under its own weight, they printed it inside a supportive gel bath. Think of it like building a sandcastle underwater, where the water keeps the structure intact until it's ready to stand on its own. Or picture pouring sand around a water balloon to help it hold its shape. That gel acts as a temporary cradle, allowing the delicate collagen to hold form as it solidifies. Once the collagen structure sets, the surrounding gel can be gently removed, leaving behind a perfectly shaped, intricate scaffold. The results? Nothing short of astonishing. Scientists were able to print complex biological structures, things like blood vessels, heart valves, and even miniature patches of beating heart tissue. Yes, beating. And here's the real kicker. These weren't just static models. The printed scaffolds were strong and flexible, and more importantly, they were biologically active. They could host real, living human cells. Cells could grow, multiply, and behave as they would in natural tissue, turning printed scaffolds into living systems. This breakthrough didn't just solve a technical challenge, it opened the door to a future where custom organs made from your own cells aren't science fiction, they're a work in progress. Okay, we have the scaffolds. They're printed, shaped, and ready to go. What's next? Well, you need life. That's where bioinks come in, specialized liquids packed with living human cells. These bioinks can contain just about any type of cell, depending on what tissue you're trying to create. Skin cells for skin grafts, heart muscle cells for cardiac repair, even versatile stem cells that can morph into whatever the body needs. Bone, nerve, liver, you name it. Once the scaffold is printed, it's time to bring it to life. Scientists either use a second printer head to deposit the bioink during the printing process or apply it afterward, carefully seeding the structure with living cells. The scaffold acts like a trellis, guiding these cells into the right places. Then comes the science of nurturing. Under the right conditions, temperature-controlled incubators, carefully balanced nutrients, oxygen, and with just the right chemical signals, these cells begin to do something amazing. They multiply, they communicate, they organize themselves into living, breathing tissue. Some of these engineered tissues have done more than just survive in the lab, they've thrived. Scientists have grown heart tissue that actually beats rhythmically, not just twitching, but pulsing like the real thing. Others have developed vascular networks, allowing blood-like fluids to flow through, which is essential for delivering nutrients deep inside the tissue and keeping it alive. This isn't just printing biology, it's recreating life's architecture from the ground up. From a lifeless scaffold to a living functional piece of human tissue, this is where the magic really begins. Real-world impact, what can we do? First, organ transplants. By using a patient's own cells to print a brand new organ tailored to their body, we could eliminate rejection risks and drastically reduce recovery time. Second, drug testing. Imagine testing drugs on lab-grown human tissues that act almost exactly like the real thing. Heart muscle tissues to test for toxicity, liver tissues to monitor metabolism. This isn't just faster and more ethical, it's more accurate. 3. Burn and wound. Healing. One of the most immediate uses for this technology is in skin repair. Scientists are already experimenting with bioprinters that can print skin cells directly onto wounds, especially for burn victims. These printed layers aren't generic. They're custom built to match the patient's body, layer by layer. Over time, they don't just heal, they restore appearance, they restore flexibility and dignity, and that's just scratching the surface. Researchers are working on cartilage for joint injuries, corneas for the blind, even pancreatic tissue for diabetes treatment. Every new layer printed is a step closer to changing and possibly saving lives. Limitations. Printed scaffolds, living tissues, beating heart cells are truly innovative. But we're not exactly at the point where hospitals can just hit print and deliver a fully functioning heart or kidney for transplant? Not yet. So what's the holdup? Let's break it down. 1. Complexity of organs. Organs aren't just blobs of cells clumped together. They're incredibly complex systems, each with its own unique architecture, cellular diversity, and mechanical behavior. Take the heart, for example. It's not just muscle. It's muscle tissue lined with blood vessels, interwoven with nerve cells, anchored by connective tissue, and controlled by synchronized electrical impulses. All of these parts have to work together perfectly, or the whole thing fails. A kidney? Even more complex. It contains millions of tiny filtration units called nephrons, each responsible for balancing electrolytes, filtering waste, and regulating blood pressure. Replicating just one of those elements is hard enough, Replicating all of them and getting them to behave like the real thing in real time inside a living body, that's a level of bioengineering we're still working toward. 2. Vascularization. Here's a big one. Blood supply. Living tissues need oxygen and nutrients to survive, and they get those from blood vessels. For tissues smaller than a grain of rice, diffusion can keep them alive. But anything bigger? It needs blood vessels running through it, ideally capillaries that connect seamlessly to veins and arteries in the body. Currently, we can print small vessel-like networks into tissues. We're talking microfluidic channels that mimic blood flow. That's progress. But physically getting those printed vessels to connect to a person's existing vascular system and stay connected without clots, leakage, or collapse, that's a whole other level of difficulty. 3. Regulatory hurdles. Any new bioengineered tissue or organ has to go through rigorous testing, not only to prove it works, but to confirm it's safe in the long run. We're talking about years, possibly a decade, before such a product could be approved for general use. We're a few breakthroughs away and getting closer every day. Here's the good news. Actual scientists in real-world labs are making breakthroughs that were unimaginable just a decade ago. Take the FRESH technique, free-form reversible embedding of suspended hydrogels that we talked about earlier. In 2019, a team of researchers from Carnegie Mellon University used it to print functional human heart tissue. Now, it wasn't a complete heart, but what they printed could beat. It responded to electrical signals like a natural heart does. It was able to mimic the motion of a living heart muscle. That might sound like a small win, but for patients who've suffered heart attacks, patches of that tissue could be life-changing. In another promising case, scientists developed a trachea-like structure using a combination of stem cells and collagen. It wasn't just a hollow tube, it was engineered to stay open, support airflow, and survive in a biological environment. In the future, this type of bioprinted airway could help children born with tracheal defects or patients needing emergency reconstruction after cancer or injury. And the breakthroughs don't stop there. Just last year, another research team used 3D printed lung tissues to better understand how COVID-19 damages human lungs. They printed a small model of lung tissue, seeded it with human cells, and introduced the virus. What they observed helped them uncover the precise ways the virus enters and spreads through the respiratory system. This kind of insight was incredibly difficult to achieve using animal models, or flat petri dish cells. With printed tissues, we get a 3D, human-specific view 1 that could accelerate drug testing and treatment development. What do these stories have in common? They're not just theoretical. They're not sketches on a whiteboard. These are tangible advances, structures you can see, touch, and test. They might not be transplant ready, but they're serving vital roles in drug testing, disease modeling, and tissue repair. Ethical questions. As we move forward, it's not just a question of can we do it, but should we? Sure, printing tissues to save lives feels like a no-brainer. But what happens when we go beyond replacement and into enhancement? Should we be printing tissues that make someone stronger, faster, or more resistant to injury than nature intended? Super muscles? High-performance skin? And what about organoids? Those tiny lab-grown versions of human organs used for research? And then there's the elephant in the lab. If we can print life, where do we draw the line? Are we approaching the territory of synthetic biology that borders on creating life itself? Or are we simply building tools to fix what's broken? These aren't questions with easy answers, and they're not just for scientists in lab coats to decide. They're for philosophers, lawmakers, religious leaders, and every one of us. What we print doesn't just affect bodies, it affects values, systems, and the definition of what it means to be human. Still, one thing is clear. Whether we're ready or not, the future of medicine is being printed layer by layer. And the conversation about its impact starts now. So, would you accept a printed organ grown from your own cells? Will labs print customized patches to heal injuries or test drugs on your personal mini liver? The revolution of 3D printed tissue engineering is already here. The question isn't whether it will change the world, it's how fast. If you found this fascinating, like the video for more stories on how science and technology is reshaping biology, and leave us a comment with your feedback. Thanks for watching. As always, stay blessed and stay curious.